This is Dr. Launen, and this is a lecture on horizontal gene transfer, specifically focusing on the process of transduction. And also because you need to understand this in order to understand transduction, I'm going to be addressing the two most common life cycles of bacteriophage. The goals for this lecture set are for you to know what a bacteriophage is, for you to know the two common life cycles and thereby classifications of bacteriophage, for you to know what transduction is in a general sense, and how generalized and specialized transduction are similar and different. So transduction is the transfer of DNA from one cell to another through a bacteriophage, which is a virus that can infect bacteria, nicknamed phage. Transduction can occur in two possible ways. It can be generalized or specialized. And it appears to be an incredibly important mechanism of horizontal gene transfer in nature, especially in the ocean and also in the human gut. So what are bacteriophage? They are viruses that infect bacteria. We also use the term phage to refer to the viruses that infect archaea. So shown on the left is a cartoon description depicting the parts of bacteriophage. Here we've got the head of the phage and the tail of the phage. And the blue is showing us where the nucleic acid found within phage, most commonly DNA, is located. This coating here is called a capsid, and it's made out of proteins, repeating units of the same identical protein. This area is called the collar, and this area is called the tail. And the tail contains what's called a base plate and tail pins or fibers. And what I'm going to point out here is that the tail pins or fibers are what first attach to a bacterial host cell. And so it is the protein recognition between these pins or fibers and the surface of a bacterial host cell that allows specific phage to infect specific host cells. The base plate is then going to dock on and kind of puncture the cell envelope of the host, and the nucleic acid that's in the head will be ejected out of the phage and into the bacterial host. To the right is an electron micrograph. This is, about half or the end perhaps of a bacterial cell, so the rest of it would be going off screen. And all these little critters are phage that are attached to and about to inject their genomes into these bacterial cells. You can see these faint outlines inside the cell, and I think what this electron micrograph is capturing is new phage that might be getting synthesized in there, although it may also just be an artifact of the image, and these may be like looking from the top down at some of these critters. That's actually more likely. All right, so you can see these things using an electron micrograph. These are really, really tiny, right? These are much tinier than a bacterial cell, which is already very tiny. So you'd have to use an electron microscope in order to see these structures. You could never see them with the naked eye, obviously, and you couldn't see them with a light microscope either, obviously, since you can barely see some bacterial cells with a light microscope. The other way that we have for visualizing phage is to infer their presence because they have blown up their host cells. So this is a Petri plate, and it is covered with a layer of confluent or continuous growth of a bacterium, and that's what this milky appearance is. So that's like layers and layers of bacterial cell that have been spread evenly over the surface of this plate. These little circles are not colonies of bacteria. They are actually holes in the bacterial growth. And the dots are where a researcher has used a Sharpie marker to count those holes. So each of these little holes is showing us that there was a phage infecting a bacterium inside the center of that hole, and then it was infecting and blowing up the host cell and erupting and going off and doing it to the neighbors. And so the infection spread out in a perfect circle, and the bacterial host cells died and were lysed, and so you get what's called a zone of clearing. It's actually called a plaque formation in the center of this confluently grown Petri plate of bacterial cells. So we can count those, and we can infer from the death of the host cell that the phage is present. All right, there are two types of phage, and they are defined by their lifestyles or their life cycles or their mode of replication, however you want to think of them. There are what are called lytic phage, which are also called virulent, and there are what are called lysogenic phage, which are also called temperate. Okay, those names mean the same thing. So the lytic phage are the simplest. They infect bacterial cells, they multiply. That multiplication causes the host cell to blow up and die, and then the bacteria or the viruses repeat this process with new host cells. The lysogenic phage are kind of a lot cooler. The temperate phage are really much more interesting in many, many ways. They infect, and then they go dormant in a stage called the prophage. And while they're in there, they're generally or thought to be, in some cases known to be, influencing the host bacterial cell. So they're essentially part of the host cell genome. Whenever the host cell copies itself through cellular replication, the prophage, which is the viral form at that moment, gets copied as well. And then something in the environment, maybe UV light or some other stressor or some signal, will trigger the viruses, the phage that are hidden within the host genome, to become evil again. They become lytic. And then that causes them to replicate, blow up the host cell, and repeat. So here's a schematic of a lytic phage, also called virulent phage, because it does a lot of harm to the host cell. You have the attachment of the phage to the host cell, and this is not drawn to scale, right? Think of that electron micrograph I showed you a moment ago. Like, this would have to be much, much tinier than the host cell to be to scale. The attachment is mediated, it's very specific. Only some phage can attach to certain host cells, okay? And that's mediated by the protein-protein interaction between the tailpins and the surface of the host cell, just like any viral attachment. So we have attachment, then we have entry of the phage DNA, and that causes the host DNA to degrade, and then the phage takes over the host cell, and it uses its DNA polymerase, and it uses its RNA polymerase, and it uses its ribosomes, and it forces the host cell, it hijacks the host cell, making it replicate and build new phage parts, including phage genome, the blue, light blue stuff, and phage protein parts, the purple stuff. These automatically assemble. There's some really cool physics in that. And then what happens is, after assembly, you get so much viral pressure in there from the buildup of the phage inside the host cell that the host cell blows up or lyses, releasing these lytic phage into the environment, and then they can go off and infect new host cells. So it's really very, very simple. All right, and this brings us back to the topic of horizontal gene transfer. So how can phage be the mechanism of transduction? And what phage do is phage can act to transfer bacterial host DNA from a donor cell that has died off to a new host bacterial cell through the infection process. Lytic phage, which I just explained, do this using generalized transduction. And this schematic is showing that. Here's a lytic phage. It's attached or adsorbed to the surface of a host cell. It's injected its genome. That has caused the complete destruction of the host cell chromosome. But the host cell machinery is being hijacked and used to build new lytic phage. And this is a bit of a free-for-all, and in this environment, in the chaos that ensues, the new phage, as they self-assemble, generally pack up their own correct phage DNA, but once in a while, by mistake, they get a little bit of the host cell genome captured in their capsid heads instead. And so this little critter here is special. It's called a transducing phage. So it can go off and it can inject that host cell DNA into a new recipient bacterial host cell. And in doing so, that DNA, this little piece of green DNA, could be integrated into the new recipient cell genome, thereby changing it, genetically altering or transducing it. This is not an infective phage, so this is kind of a dead end from the point of view of the phage or virus, but it is just simply a vehicle for transferring bacterial DNA from this donor cell to a new recipient cell. This is a very low efficiency transfer, but it does and can and has worked many times in nature. All right, I'm next going to go on and talk about lysogenic or temperate phage and how they participate in something called specialized transduction.